Good afternoon, everyone. The theme of the symposium is about modeling the dependence in the imaging studies. And during the last talk, we heard how to handle the dependence in the longitudinal studies. During this talk, I'm going to talk about another kind of dependency that is induced accidentally or by intention by recruiting the related individuals from the families. Here's the content of my talk. In the first part, the familiar dependence is considered a nuisance for the covariate inference. And in the second part of the talk, I'm going to describe how this familiar dependency can be used to perform advanced analysis like heritability. The main motivation for this work is the Human Connectome project. The HCP uses a cutting-edge MRI technology to gather structural, functional MRI data, followed by a comprehensive set of non-imaging phenotypes. What I'd like to emphasize here about the HCP is that it uses extended twin design, and hence the individuals in the HCP, they are related, and they are not independent of each other. And this level of relatedness between the individuals in the HCP is captured in a metric so-called kinship matrix. Here, what I'm showing you is the toy example, an example of HCP design, 10 subjects from 4 families when the level of relatedness are color correlated. And for instance, between the MZ twins, they are expected to share 100% of their genome. Also, in studies of completely unrelated individuals, there is another sort of weak dependence can be defined, and this is captured in a genetic relationship matrix, or GRM, where each element of the GRM is simply a Pearson correlation between the 2 subjects of minor allele frequencies among all of the individuals. And during the rest, and towards the end of the talk, I'm going to describe why this GRM is important and how it can be used. We are using a linear model to find the areas in the brain that their structure or functions are related to a covariate. And in the context of GLM, the main assumption is the independence. But clearly in the HCP, the subjects are not independent of each other, and ignoring this assumption may lead to a spurious finding. To see that how these spurious findings could be severe, we performed a null simulation using the HCP S1200 data release kinship matrix. And there is no covariate effect in this simulation setting. And we are comparing the observed p-values with the theoretical ones. What you are seeing here is that these shaded areas are the lower and upper bound for the confidence intervals, and the red dots, are the observed minus log 10 p-values. If the assumption is valid, if model works perfectly, we would expect to see that these p-values, these red lines, lie on the shaded area, which is clearly is not happening, as indicating the inflated false positive rate. So what's the solution? Linear mixed effect model is a standard statistical method to perform the covariate inference in presence of the kinship matrix. What we did here, we look at the relationship of the reading English ability between all of the free surfer phenotypes from HCP S1200 release, and we compared the test statistics of the OLS when you ignore the kinship matrix, and you cannot take into account, with the test statistics that comes from the linear mixed effect model. And you can clearly see here, ignoring the kinship matrix inflates the false positive rate. And it's not only with the reading ability, we see the more severe results if we look at the relation between the IQ and compare the test statistics between the OLS and mixed effect model. So the take-home message is that using the OLS with the HCP subject inflates the false positive rate. But there is a gap here. A standard imaging software cannot take into account the kinship matrix for the covariate inference. On the other hand, tools that in the quantitative genetics field cannot provide a spatial statistics like cluster size or TFC inferences, nor multiple testing correction. More importantly, these tools are not scalable because they're using a numerical method for the parameter estimation and inference, which is prone to the convergence failure. And parametric inference is only valid asymptotically. Hence, there is a compelling need for a computationally efficient mixed effect model that can do the standard analysis in the imaging community. What we did here, we compared the 3 software packages that can accommodate the kinship metrics for the covariate inference. The first package that we are using is a R package of LME-kin. It implements the linear mixed effect model, which is the best statistical practice. As I mentioned earlier, it relies on the numerical method for the parameter estimation and inference, and it's interactable for mass univariate approach. The second package that we are comparing is PALM, which implicitly models the family relatedness by permuting the subjects within exchangeable blocks and can give you the FWE corrected p-values. The last package is our recent work that we call it Ninja, and the method is implemented in the solar eclipse. And it provides an approximate solution for the mixed effect model maximum likelihood solution. I'm not going to go through all of the math behind the Ninja, but in summary, the way that Ninja works projects the data and the model, mixed effect model, to a large dimension space using the eigenvectors of the kinship metrics as just for the fixed effects. And after that, it suggests to build an auxiliary model and use the regression line to estimate the random effect and the heritability. And also another advantage of this projection is that it allows us to do a fast association or covariate analysis. In order to compare these 3 software packages, we use the simulation. In the first simulation study, we did the null simulation. Again, there is no covariate effect, and we look at the false positive rate of the OLS when you ignore the kinship metrics, Ninja, and PALM. And what you are seeing here is that clearly ignoring the kinship metrics based on the level of the dependence, based on the level of variance explained by the kinship metrics, inflates the false positive rate, while both Ninja and PALM, they are valid. We did the second simulation study, and we compared the power of the between the Ninja and PALM. Again, they are both valid, and their performances are similar. It's not only with the simulated data. We see the same things with the real data analysis. Here, we are comparing the uncorrected p-values between LMER, PALM, and Ninja using the 68 cortical thickness regions from S1200 release and their relation between the reading ability score. And what you can see here is that Ninja and LMER, they are the same performances, and they are slightly more powerful than the PALM, and there is no surprise because Ninja provides an approximate solution. And that's the same thing if we want to compare the FWE corrected p-values. We did the same analysis, but this time we performed the association of we did the vertex-wise analysis. What you are seeing here is the areas in the brain that they pass the 5% FWE vertex-wise analysis. To get these maps, Ninja only requires 2 hours for 5,000 permutation, and PALM does the same analysis in 5 hours. Hopefully, by now, I could convince you that if you are using the HCP and you are doing the covariate inference, you need to use the packages that they can accommodate the kinship metrics. In the rest of the talk, I'm going to talk about how this familiar dependency can be useful to do more advanced analysis. Tools that are available for doing the heritability, as I mentioned earlier, they are relying on the numerical methods. More importantly, they can be conservative. On the other hand, Ninja can give you a fast and powerful analysis. What you are seeing here is the vertex-wise heritability estimates of S1200 cortical thickness of the HCP. If you want to use the solar to get this map, you need to wait for 4 hours. On the other hand, Ninja does the same analysis in 1.4 seconds. And to get the FWE corrected vertex-wise analysis with 5,000 permutation, Ninja can give you this map in 2 hours. Remember at the beginning of the talk, I was talking about this weak dependence between the unrelated individuals using the GRM. When we have completely unrelated individuals, we would expect that the values of the GRM should be 0. But clearly what we are seeing here are that they are not all of them are 0. Here we use a different data set, 300 completely unrelated individuals, and we look at the heritability estimates of the FA values that comes from the TBSS and 42 ROIs. And what you are seeing here is the box plot of the heritability estimates, and all of them clearly not 0. So it can be useful. Recently, mixed effect model has been suggested for the genome-wide association of analysis of the unrelated individuals. And the tools in the genetic field to do that, they are not scalable, not surprisingly. To best of our knowledge, the fastest implementation is FastLMM. And for doing the association of 22 ROIs with more than 1.5 million SNPs, FastLMM requires more than 700 minutes, and Ninja can do it in 2 minutes, which this is because of the reduced computational complexity of Ninja. We use the Ninja to do the genome-wide association analysis of more than 2,000 phenotypes in the HCP with more than 9 million SNP using the all HCP analysis. And what you are seeing here is the Manhattan plot of the p-values. The red line here shows the usual p-value for the GWAS, and the blue line is the 5% FWE threshold corrected for more than 20 billion tests, corrected for the number of tests and ROIs in the image. And to get this Manhattan plot with 1,000 permutation, only need 10 hours. So in conclusion, for the covariance inference, we highly recommend to use the software packages that they can accommodate the kinship metrics to get the valid inference. And also Ninja can provide a computationally efficient linear mixed effect model for the genetic analysis of the imaging phenotypes and gives you the FWE p-values. Thank you very much for your attention.